first natural science seminar of the semester. This is it. And a lot of you are getting extra credit. Some of you are laboring at real credit, like if you're in one of my classes. So we do that because we want to give you a real solid reason that will benefit you as well as benefit us for coming to Natural Science Seminar. So that's how things are working there. Today, we don't have one of those boring professors who's going to talk on and on and on and on about things that only two people in the audience, Dr. Bradovich and somebody else, can understand. Okay? What we're going to do, what we try to do a lot is send students out to do exciting things in science and then call them back to give a report on what they did. We have two examples of that, and the call you back might be a little muddled in one example, but we sent Rachel Hubbard over to Western Michigan University over the summer. This last summer? Yeah, boy, it seems like an eternity since then. So she did some research over at Western Michigan University in Kalamazoo. There's a song about that city. And it's fun if you're my age, you kind of remember the reruns. I'm trying. Then we have a group of 3 students who did research with me on chemical topics, and some of them are really old and some of them are younger. So the idea there is to give you a little picture of how science actually works, because it's usually a senior scientist training younger scientists, and we have that model in effect here. I'm the super old scientist who designed the whole project, and then we have successively less super old people who do the actual work. And that's really how science works. There's a guy at the top who figures out what we want to have done, and this applies for Rachel's experience as well. And then there are people underneath that person who actually do it. And then everybody gets together and they write a poster, which has been done for both people, or they write a paper, and then it gets picked apart and they do edits, and it gets into the scientific literature forever and ever. So, without further ado, because Dr. White is very impatiently waiting to hear from important people, let's, who's going first? Okay, we got the team of 3. We've got John and Sam and Kristen. Thanks. Yeah.
So, I'm the second old person. My name is Kristen Lewis. I graduated from here last May with a degree in math, and I am teaching under Dr. Baldwin here while finishing up a second bachelor's. And so I am working directly under him, and I've been working on this project for about 2 years. And then John and Sam have come in and helped me with various parts of the project. And what we're trying to do is make some of the reactions that we do in Dr. Baldwin's class greener, or more environmentally friendly. Most reactions take a lot of work and use a lot of things that we end up throwing away at the end. And so we were trying to take that out and only use what we need to make the product with no waste. And when you look at combining things, there's 4 different ways you can combine 2 reagents or 2 chemicals. You can have a liquid chemical and a liquid chemical. And usually those are pretty easy to do. Liquids mix really well, the reaction can start right away. So we kind of looked at that and said, it had already been published and it's not very challenging. And since, especially since it had already been published, it's going to be hard for us to do something we can publish that's anything new. And so we're like, okay, let's try a solid and a liquid. And so the first one that I did as a sophomore in organic for my 3-week project was benzoic anhydride and benzylamine. Benzoic anhydride is the solid, and that's this one right here. benzoamine is the liquid, and I was trying to make this amide. And you'll notice that everything we try to make is an amide in this case. Next. Normally, we use some sort of catalyst to make the reaction go a lot faster. So when I first ran it, I ran it with the catalyst. I also heated the reaction. That usually helps get it going. If you have been in chemistry, you've heard of activation energies. Heating sometimes lets you overcome that activation energy and the product will be ready to make. Then in organic, we've got to wash out all the stuff we don't want. In a lot of symmetric anhydrides, you're going to have a piece of it that's going to end up off by itself. That's usually called the leaving group. And you got to get rid of that. You don't want that in your product. And then we hardly ever get every single molecule of every chemical that we put in is going to react to make our product. So we also have to get rid of any extra starting material. And usually that happens with an acid wash, a water wash, and a base wash. You do the acid wash to get rid of any basic materials. The water helps remove anything else that we might have, in this case, protonated, and it didn't get out, and then we do an acid wash. And to do that, we take our sample and we dissolve it in a liquid so that we can really mix liquids well together. And that's where the organic solvents come in. And we got a pretty decent yield of 66%, which means of all the stuff that we put in at the beginning, 66% of it made product. And the crystals that we got were yellow. So we're like, okay, can we get rid of the catalyst? If we can get rid of the catalyst, then we don't have to get rid of that at the end. Catalysts go in and come out of a reaction exactly the same. So if how much catalyst you put in, you got to get that much out of your product. So let's try to get rid of the catalyst. Heating requires electricity or gas or combustion. That's another source of something that we're consuming that's not in our product. Let's try to get rid of that. I wonder if we can control how we put the reagents in so that we only have to do one, maybe two types of washes. If I carefully pick the ratios of what I put in to the beginning, maybe I only have acids that I have to get rid of at the end. And if I only have acids, then I only have to do a base wash. And organic solvents usually never become part of your product. So let's try to get rid of that. And so for the green reaction, that's what we tried. We used no catalyst, we used no heat, no organic solvents, which means when we go to do our wash, we're going to be putting an aqueous liquid on our solid product. And that's kind of a gamble because you don't know how much of it is really going to get washed. But we got a 98% crude yield. We got better results, or at least more results at this point, by not using any of that extra stuff. And our crystals were white or colorless, which is usually another sign that they're better quality. And so now we had to see, okay, did we actually make something better? So now we had to do data analysis. And if you've been in organic chemistry, you'll recognize IR, TLC, and melting point. If you haven't been, melting point is basically the point at which it melts. You know, when we think about ice freezing and melting at 32 degrees Fahrenheit or 0 degrees Celsius. But it's not actually at that exact point. There's usually a range. So for our range here, it was 104.6 to 105.6 degrees Celsius. The smaller the range, the more pure your product. Usually we want to get below 3 degrees, separating the two for a pure product. And we got 1, so that was great. Then the TLC lets you know how many different things are in your sample. So when you look at the number of spots, that tells you how many things are in that sample. And in this one, we've got 3 different lanes, which might be hard to see if you haven't done TLC before. We've got two spots here that are right above each other. That's one lane. We've got one spot that's kind of in the middle, and then one spot that's on this side. So what we did is we took the 2 starting materials, and those are on the outside. The benzylamine and the benzoic anhydride are on the outside. And then in the middle is what we made. And so if what's in the middle doesn't line up with any of the spots on the outside, we know that we don't have any starting material in our product. And that's what we see here. We have one spot, which means we have one thing in those crystals, and that one thing is not a starting material. So then we have to go, okay, did we make the amide? We know we made one thing, we know that it's pretty pure. But did we make the amide? That's when we go to the IR. The IR lets us see the bonds in between atoms. So if you look at the purple, that little peak represents a carbon atom. And then the O represents an oxygen atom. And we've got a double bond between the carbon and the oxygen. That we see at 1637, the purple colored peak. That lets us know that we have a carbon double bond oxygen. And what's really cool about carbon double bond oxygens is depending on what's around them, that affects where they come out. And so we can say that that carbon double bond oxygen is part of an amide functional group, which is carbon double bond oxygen next to nitrogen. And that's what you see up there. Then the nitrogen and hydrogen, the bond isn't drawn, but the IR tells us about the bond, not about the atoms. That is shown at 87. And that lets us know we have a bond between a nitrogen and a hydrogen atom. And I'm not going to go into detail, but we also don't see bonds that correspond to the starting materials. So now we can say we made something that's one thing. The melting point range, the TLC, and the IR tell us that it's pure, and the IR lets us know that we did make an amide. So that was my 3-week project. 3-week projects never work. Never, ever, ever work. But this one did. So we thought, let's see if we can go farther. And we went and tried to expand this to take it to Indianapolis. And that's where John's at.
All right, so I'm John, and I'm the younger chemist of the group, the youngest one actually. And like Kristen said, 3-week projects almost never work. Mine didn't very well, and so this was like my second experience with research, and I collaborated with Kristen on building off of what she had done with her 3-week project, and she really took me under her wing. So we looked at that first reaction, and that worked so well, and we did it again, and we had success, and then moved on to some other combinations of liquid, solid, amines, and hydrides. So we had some more success with this reaction. You can see that the melting point range is in the lower left-hand corner of that picture. It's only about 1 degree, which indicates that the sample is very pure. Also, if you just take a look at the picture, like the crystals, they're so like clear and nice. Like we were really happy and excited about that. And sometimes just by looking at it, you can tell like if it's a good pure sample or not. So we were excited about that. So we moved on to another reaction. Again, you can see just from the picture that the crystals are pretty clear. We were really happy. However, this reaction we had a little bit of struggle with at the beginning. And the reason why is that this product right here has some acidic properties to it. And when we used the procedure with the base wash, acids and base react, and so it really messed us up. So what we did is we made a small change, and we used an acid wash to get rid of any impurities in our product and sample, and ended up with a huge success. And then we moved on to another reaction, and this time we used liquid anhydrides with solid amines, versus before we were using solid anhydrides with liquid amines. All right, going for the gold. So we were a little bit like shaky about this. You know, will it work if we switch up the liquid-solid combination? And to our happily surprised, we were really excited because this reaction worked really well again. We were beyond excited at this point because everything had been working so well so far. Each reaction, though, did have its unique characteristic to it that we had to work out, but we were able to get most of them to work really well so far. You notice that on the picture, the crystals are a little bit more yellow, and the reason for that is the starting amine was a little bit, it was yellow in color. So that's why the product turned out that way. The melting point range, you can see another just 1 degree, and so that indicates some purity, and then the IR corresponded with the amide peaks that we had desired. So really happy again. Moving on to the next reaction. This is where it started getting a little bit frustrating. You can see just by looking at it, you know, the crystals just don't look as good as what they were before. Also, you can take note that the melting point range is a lot bigger. Like it's a 234 to 245. That's a lot more than just 1 degree. So there's probably going to be impurities in this sample. So that was frustrating. So we're like, okay, we'll move on. We'll try another one. You know, this one. You can see just like looking at the plate, like we're like, really? You know, those are not good crystals. That's not what you want. You know, there's probably some product in there, but there's probably a lot of other garbage that we just didn't want. So the melting point range again, pretty big. It's not what we were hoping for, but overall, we experienced some success and then some frustration throughout the whole process of doing this research, and learned a lot, and we're pretty happy. So.
And we worked on these last couple reactions, and we got to the point where working on it anymore really isn't worth it. We haven't been able to figure out the problem. We have 4 reactions that worked really, really well. Let's try to finish this grid that we have here. We didn't touch the liquids in the liquid combination. We got good results for solid liquid and liquid solid. So what happens if we try solid solid? That's where we were actually always trying to go, because solid reagents are a lot safer for students to work with, and they're a lot easier to work with. So if we can find a green reaction that works to make a variety of amides, that's going to be really good, and that we can publish. So for my thesis, I'm working on the solid-solid, and Sam has been helping me with that. So she's going to talk about her research.
So for my independent study, I decided to work with the solid-solids. And we started with benzoic anhydride and the yellow substance, the stilbene, that they used for their liquid solid combination. And we went through the process. We started with we started with just grinding the solids together. So we wanted to green it completely. No solvents, no heat, no anything that could minimize the greening process. We just used a base wash so that we weren't using the added acid, and it didn't work at all. We had no product. So we went back and tried using just solvent, and that didn't work either. So then we went back and tried just using heat. We finally got a product, but our results were inconclusive. We couldn't prove based on TLC and IR that we had product. It was getting better as we went, but it wasn't like provable. So we added in things, and it was just very inconclusive. So, failure. Get used to it. It's okay to fail sometimes, because failing tells you where you're not going, and it tells you what to try next. And you know, every project you're gonna have is gonna have some element of failure in it, unless you just get really, really, really lucky.
For example, the work that she did told me to not work with the still bean. Let me try something else. And so the next reaction that I tried actually worked like a charm. And we got a great IR, we got a great TLC, and her work helped me know where I shouldn't put my time so that I could find something that worked. And so a lot of you that are in inorganic or organic, you're going to be doing 3-week projects at the end of the semester. So we wanted to let you know, sometimes you get lucky and it works. Sometimes the professors have a great idea, but it doesn't work. So if it doesn't work, that's okay. You're helping the next generation of students know what not to do. And with that, we're going to turn it over to Rachel and let her tell you about her experience at Western.
John? Woo!
Go Seahawks.
Wrong place. So yes, as Dr. Baldwin said, my name is Rachel, and I'm a senior chemistry student here at Spring Arbor. And I did have the privilege of going to Western this summer to participate in the RU program. And I want to explain what that is. So it stands for Research Experience for Undergraduates. And it's a program that a lot of different colleges and universities will host, and it's funded by the National Science Foundation. It's a 10-week program in the summer, and there's different disciplines that are involved with the program. I wanted to, because I know I found out about it when I was a freshman, but I didn't know what it entailed. So I want to tell you about how there is a stipend. So these can fluctuate between the different universities that host the RU program. But for me personally, it was a $5,000 stipend, so that means it didn't cost me anything, but they paid me to go. And also they covered housing, and they covered, I think, like 2 meals a day and then like 1 on weekends or something like that. And the RU program will reimburse students for travel if they live far enough away. And so for Western, the application deadline is March 4th. So if you're a sophomore or a junior and this sounds interesting, then you need to apply sooner than later. So each institution, so like mine was at Western, they choose 10 of the applicants, and these pictures show all 10 of us together. This is me, obviously, and this is Haley Marshall. She's also from Spring Arbor and was able to go. And from those 10, we're all placed with different professors or mentors. So my mentor was Dr. Shimshina. I was the only undergraduate working with him. And it can fluctuate depending on which mentor you receive. But for me, in my experience, Dr. Shimshina poured so much into me. He spent hours and hours with me in his office, in the lab, teaching me about proteins and genes and primers, and talking about primers again because it's really confusing. And even at the very end of the summer, we went to Notre Dame to do a proton NMR on a protein that we've been working on. And he took me and one of his graduate students, and I went basically just for the experience. He was like, do you want to come just to see this life? And so it wasn't just him and I. He has 3 graduate students in his lab. He has Harry, Hamide, and Nafise. Harry's from Kenya, and Nafise and Hamide are from Iran, and I still keep in contact with them. And this is a picture kind of showing a little bit of what his lab looks like. And it kind of wraps around the other side too. The thing at Western, well, the next slide will show. So what I learned is I got to taste a taste of what a bigger school looks like. And not only just in the expanse of it, but also it's a very research-driven school. And so there's a building, we were in Heineke, and that building is devoted to labs for professors who do research. So that building is only for the researchers and their graduate students. I also got a taste of what graduate school itself looks like. So it was like being a fly on the wall, but also getting to participate, because they really welcomed me into their family, because that's really what it's like. And so you have the graduate student and their mentor, and so I got to see what their relationship is like, what the hours are like, what the workload is like, and also biochemistry, because I told you I'm a chemistry major, so I really didn't know very much about biology going into this. And Dr. Shimshina knew that when he picked me, and he taught me so much. There's also, through the RU program, our first week there was like different seminars every morning, and we would learn different things like ethics and safety and different professors' research that are there. And then we would also have them every Friday for the remainder of the 10 weeks. And one thing I wanted to, just like one bit of information to share, is that they taught us that science is no longer vertical, it's horizontal. So that means that, like for me as a chemistry major, I might think that chemistry is the greatest and all these other things don't matter. But it's not true, because there's a lot of bleed over and it spills into one another. And so I want to encourage you guys, if you're in a support class for your major, to really apply yourself, because that will even show up in your higher level classes here, and they'll come into play, and so they're all interconnected. So that's telling you about what the RE program is like, and what kind of a little bit of what my experience was like. And so now I want to tell you about the research I did. So each of the 10 students had a different research project, and we had to do the research and also write a paper and give a 20-minute presentation to the other RU students and their mentors. So this is mine. So my research was bioinformatics and gene expression of Tanapox virus proteins. So what is that? Tanapox is a pox virus. You might be familiar with like the chicken pox. It's in that family. It's double-stranded DNA. It was discovered in Kenya. And this particular virus has predicted to have 156 different open reading frames, meaning that it can potentially encode for that many proteins. It infects both humans and primates, including the rhesus monkey. So some unique features of Tanapox. It's non-fatal, which is good. It's transmitted through things like mosquitoes and direct contact. When someone develops this, they get like headaches, fevers, and then anywhere from 2 to 10 of these lesions can occur. And the majority are in adults, and it takes about 6 weeks from when you first get it to when it heals. Now, through my research and figuring out like why is Dr. Shimshina interested in this? He's interested in Tanapox not so much for the danger of Tanapox, but because it's related to more dangerous viruses. So smallpox and monkeypox are extremely dangerous and can be fatal, and Tanapox has similarities to these. And so by learning about Tanapox, we can draw hopefully some conclusions or gain a greater understanding of these more dangerous ones. Bioterrorism is something to think about. That's the possibility of someone trying to infect a large group of people with one of these viruses. International travel brings up the issue if someone were to go to a different part of the world and bring back a virus, and we would need to be able to know, to be able to recognize it, know what it is, and how to treat it. And there's a professor at Western who, Dr. Asani, who has worked for like 20 years with Canapox, and he is looking at one of the proteins potentially being a tool for oncolytic therapy, which has to do with cancer. And the reason why we're looking at proteins in Canapox is because they can provide a window to see what the life cycle is in the viruses and help us to design new therapeutics to try to treat it. This can be illustrated by an example. So in us, we have a poly-A binding protein, and it will bind to the poly-A tail of our human mRNA. This will recruit the ribosome to come and translate the RNA into a protein. Now, in the rotavirus, which is separate from Canapox, but it serves as an example, it has a protein called NSP3. It does a very similar role where it'll bind to this 3' end and recruit the ribosome to come and translate the virus's mRNA. Now, knowing this fact about NSP3's function and role, it becomes a potential target because if we can block or inhibit that binding here, then that breaks down this part of the virus's life cycle. And if we do target NSP3, it's not going to harm humans because we use a completely different protein for that function. So knowing the importance of proteins in viruses, this is the list of the genome of Canapox. Each arrow represents a different open reading frame, which means these are all areas that can potentially, it's the information required to make the protein. And in the first month of my research, I was actually doing research on the computer, and the remainder of the time I was in the lab. But for the beginning part, I looked at the first 52 of these proteins, and it took me a month because I extensively studied each of the proteins, trying to gain information on them. We hypothesized that by using bioinformatics techniques, we can identify currently structured and potentially functional domains within Canapox proteins. So in proteins, it can form these like globs, I guess, that can perform different functions. And a lot of the proteins and most of the proteins in Canapox, nobody knows where these regions are. And so that's what my research was, to try to find these. So it's like searching for them. In each protein, you have the primary sequence, which is the string of amino acids. And based on the order of that string, or that like long chain, it can direct protein folding. So that's where the secondary structure comes in, because you get these folds in the protein. And tertiary structure is how those folds arrange themselves. The folds are alpha helices and beta strands. And when these come together, they can form a blob where you have hydrophobic amino acids on the outside that hate water, and the hydrophilic amino acids on the outside that love water, and that's why they fold in like that. And these are the domains that we were looking for. This is an example of a protein that you can see it has 3 distinct domains. And how I was finding those. So I was trying to find them by looking at the primary sequence of each of the proteins, because I was given a list of the proteome of Canapox, which lists the sequence of all of those 156 different potential proteins. And I used these are all different computer programs. I looked for secondary structure, tertiary structure, and I also looked at things like solubility, which gives a key. If it's not very soluble, it's hard to work with in the lab. So that was a key thing. And also PI number relates to the affinity that a protein might have for nucleic acids. And that's what we were really looking for. We were trying to find proteins that potentially bind to nucleic acids. So at the beginning of when I would take a protein and I would try to figure out where these domains are, I would put it in a program on the computer called SMART. And this is what, for the protein 30L, I got results that the gray line represents the amino acid chain, And these orange boxes, they don't really mean anything to us. It just means like a disordered region, but they weren't specific domains. And SMART tells us, is there any known domains in this particular protein? So SMART said no, but that's just what we think. So I use a different program called Protein Blast, which takes the primary sequence of the protein I'm looking at, and it compares it to all other known proteins in this big data bank. And what it told me is that from amino acid 10 to 115, 10 to 115, the protein in that stretch is similar to these different, it turns out the Plasmodium are parasites, they're malaria-related parasites. And also from 5 to 176, this particular protein is similar to all these different pox viruses, which include the monkeypox and smallpox, and they all have unknown function, which means that it doesn't mean that they don't have function, it just means that we don't know yet what it is. So this told us that potentially there's areas in this protein that are similar to all these other ones. Maybe they have a similar role, maybe they have similar structures. The secondary structure prediction, it would tell me if it thought it would have, based on the primary sequence, if the program thought it would have alpha helices or beta strands and how many they would have. There was GlobPlot, which predicted the globularity or how the domains form, how they form these chunks. And this green line shows that for 30L, it was predicted to be entirely globular, the whole protein. Tertiary structure, there's a program called FIRE that would take the predicted secondary structure of each of the proteins I looked at, and it would compare it to other known proteins and their secondary structures and try to make a connection between if they had similar secondary structures, maybe they fold in the same way. And based on that connection, seeing what those similar proteins, what their function is and how they behave. And maybe that would give us clues into what the protein I was looking at, how it behaves. From those 52, Dr. Shimshina, Dr. Asani, and I sat down and we looked at all the different data that I had gotten, and we tried to find ones that we thought would bind potentially to nucleic acids. And you can see from the 3L, 30L, and 31R that they were related to different DNA type nucleic acid binding proteins. And they also have secondary structure and are predicted to be globular, which is good because we were looking for structured domains. Not every domain is structured, but that's Dr. Shimshina is a structural biologist. So that means he tries to determine what the proteins look like, which is key to knowing how they act. And from this information, we moved into the lab. So we started with that idea, those 3 proteins. And this is the point where I moved into the lab and got to actually work with the molecular biology. We had an expression vector. So we took the genome of Canapox, we put it in a PCR machine with primers. PCR means polymerase chain reaction. And basically, we created these primers that would attach to the specific region of the genome that we wanted, that has the information needed to make the protein. And we would basically copy that and make a bunch of these inserts. And once we had the insert that contained the information we wanted, we would stick it in this expression vector, which is just a piece of DNA. Then once we got the insert, purified the insert, cut the insert so that it could connect to this, it was put together. It has a histidine tag there. So that means when the translation occurs to actually, the transcription and translation occurs to make the protein, this histidine tag is going to be connected, which is going to be important later. So now that we had the plasmid, which is the complete circle with our insert, we need to make more of it. So that's where we put it into E. coli and let the E. coli do the work and keep reproducing. And once we had the amount of plasmid that we wanted, we killed the E. coli and harvested the plasmid. And we initiated transcription and translation. And that gave us our protein. Once we had the protein, we needed to purify it. And that histidine tag, which histidine is an amino acid, and because it became a part of the protein, it was here on the end, and we were able to purify it with these beads that have nickel on them, and histidine has a high affinity for nickel. And so these would connect, leaving all this other unwanted stuff to wash out. Now that we had the protein, this is the part where we went to Notre Dame, and from the picture with Dr. Shimshina at the beginning with that huge NMR machine, this 800 megahertz machine, it was like freezing in that room because it takes so much to cool the machinery. And this, I believe Dr. Shimshina told me that because there were peaks in this region, it indicates that the protein that we have is structured. And when I left, this was like the very, very end of my time there. And he was encouraged by this result to continue doing research with this particular protein. And so once you even get something like this from the NMR machine, you still have to determine what its structure is. And that can take like up to 2 years or like, I don't know, more, but it takes a really long time. And then once you get the structure and you know if it has alpha helixes or beta strands and how many and where they're at and what position they are relative to each other, once you have that, then you have to figure out what the function is, because there can be like binding sites and how it can fit with other things. And also what amino acids are involved in the DNA binding, what portion of the proteins involved with that. And then finally, through all of that, to eventually get to a therapeutic, so we can try to find, like at the previous slide where that NSP3 protein was so important in the life cycle, if we can find proteins like that, and especially ones that bind to nucleic acids, we can try to prevent that binding and hopefully destroy the virus from replicating in our bodies. I want to thank my mentor, Dr. Shimshina, and his graduate students, Dr. Asani, who provided so much insight into Tanopox, and Dr. Spitzbergen and Team Stapleton are the people who run the RD program at Western.